to make these stocks in the freezer. And we're working together with companies, including this one, who are investing in building modular manufacturing plants that sit in shipping containers, and in which all the media and buffers that you need are present there in a dried form. The only thing you have to add is utilities, water supply and power supply, and you could churn out the first batches, first thousands of batches, in a matter of a couple of weeks. We haven't demonstrated this yet, but the potential is there to do this, and so we're working very hard at the moment. Also engineering the antibody format that we produce to make it very compatible with yeast, to hopefully, when next outbreaks come around, to be able to respond much more rapidly than we could today. I have maybe 3 or 4 more minutes to go, and I want to introduce something which I have never talked before in public, and so you know, after 35 years of recombinant protein production, probably closer to 40 years now, it's still really trial and error if you have a new protein to figure out whether it will express or not. And illustrating that, although we know all of these components in the secretory system, our integrative view of how they work together is clearly not sufficient at this point to generate predictive models of whether a particular sequence will fold in the plasmic reticulum. And so Morgan Bone, who is now just graduated and is with Peter Walter now at UCSF, she has bravely started to tackle this problem by generating a new method, not to study secretability of 1 or 2 model proteins, but to try and generate secretability data across the proteome. And so she's worked for 5 years to design a new type of cDNA library where we are generating domain-sized protein fragments across the proteome. And we're focusing on single domain-sized fragments, because if you have a big multi-domain protein, the presence of a single domain that doesn't fold will kill the secretability of that protein. So we chop it up in domains. And what we then use is yeast surface display as a proxy for productive passage of a protein fragment through the eukaryotic secretory system. So in yeast surface display, you fuse the protein at its C-terminus to an anchor protein that by nature is incorporated in the outermost layers of the yeast cell wall. And so the protein entirely transverses the secretory system, and because the fusion is at the C-terminus, the impact of the C-terminal fusion partner on the folding of the N-terminal part is relatively minimal. So the trick was here to really be able to capture the entire human proteome into a yeast surface display library that had just a few million to a few tens of million clones. And so that is technical, and I will skip it, although it's very important. So she designed an entirely new cDNA preparation method which incorporates normalization of the cDNA library in combination with random priming and starting from fragmented messenger RNA. And so this has now generated a library that overlaps the size range of human domains. We would like to get this a little bit bigger still. So the median size of an independently folding human protein domain is in the neighborhood of 85 amino acids. And so that's what we're trying to cover. So then we sort these libraries, we induce expression, and we sort these libraries using a combination of magnetic activated cell sorting and fluorescence activated cell sorting for those protein fragments that show evidence of being secretable. And so from about 2 million clones, so our proof of concept libraries are about 2 million clones, we can get expressibility data. So we do this in triplicate, and we only consider something secretable if we see it secretable in the 3 different replicates that we have. So from 2 million, we can get expressibility data of about 150,000 protein fragments, hitting about 25% of the entire proteome at least once. And so we now have to extend this, amplify it maybe 25-fold or so, to really get to very decent levels of coverage of the human proteome. So what this allows you to do is to generate catalogs, databases, where any of you could go and have a look. If you're going to express a particular part of a protein, and you want to make an antibody, for example, that recognizes a conformational epitope, you could go into the database and get experimental evidence on whether you can actually make that. So other applications are, of course, more fundamental in nature, where we've seen now that a substantial fraction of what we see has a PDB hit, and that allows us then to do data mining on a structural level to see which features of proteins are enriched in those that can be secreted versus those that cannot be secreted. And sometimes you come up with very peculiar cases, like these are two different members of the same protein family. This one is non-secretable and that one is secretable. And that now through data analysis, data mining, hopefully will allow us to extract rules and predictive models of what we can make. Perhaps that's naive. We will see in a few years how far that will go. So we're collaborating with groups that are in deep learning. These are the new methods that are used in facial recognition in self-driving cars, right, to do feature extraction, automated feature extraction, non-biased by any insight of the human mind, unfortunately, to see if we can see patterns there and predictive value. So that is my group, a very, very good group at the moment, working on biopharmaceuticals and bioanalytics, but always starting from basic research and building our own tools. I think that's something that many of us here in this audience share from Gobin's legacy in the beginning. So this is Gobin's at a retirement ceremony we had for Walter Fierce in 1997, exactly 20 years ago now. And what I recall, I was a grad student at that moment. The secretaries who were organizing this meeting, they told me, the person, there were a few prima donnas that came, who were very hard to, we had a lot of work with them. But Gobind, he came in economy class, so his ticket was the cheapest of anybody of the people who came. And we had not a single problem with him at all, It was just, that reminded me, and when I then met him finally in 2000, I could clearly relate to that and understand what they were talking about. So thank you very much. That was all I had to say.
Thank you for this inspiring talk. Questions? No question? You don't want to really investigate any of his libraries? Oh, yeah.
With this new delete strain, you would of course put some human-like glycosylation on proteins, and that would make the proteins more stable, presumably by making them less amenable to proteolytic degradation.
Yeah.
But would you necessarily replicate the natural glycosylation that's necessary for folding to occur?
Yes, so that's exactly what we do.
And also the antigenicity that might be involved with some of the glycosylation if it is non-human, if the enzymes end up putting things a little different.
Well, apparently I didn't make myself clear all too well. So what we did is exactly mimic the glycans as they are found on the human IgG that is in all of our blood. So the sugars that we have on there are exactly the same ones, except for this proportion of single glucnac that we generate now by doing the glycodelete engineering. Yeah. But that, in fact, if you look in the proteomics data sets that are there today in the databases, you do find glycopeptides that have single glucnac, and they originate in lysosomal degradation of glycoproteins. And so this is not as unnatural as you might think in the first place. And so the glycans are not only important for stability, they're also very important on antibodies for triggering Fc gamma receptor dependent effector functions. And so yeast does not make core fucose on those glycans in the first place, so you don't have to engineer it out. And if you have non-core fucosylated glycans on an IgG, the antibody-dependent cellular cytotoxicity effect goes up by a factor of 100. And so if you have virally infected cells that need to be killed off, or tumor cells that need to be killed off, that's exactly the glycosylation profile you want.
Any more questions? Yes.
Why choose Pichia instead of the Saccharomyces?
Right. Well, so Pichia pastori is a metallotrophic yeast, and there's a few reasons to use it. First of all, its N glycans by nature are much shorter than those of Saccharomyces. So if you start engineering it, the impact on the physiology is smaller than if you do that in Saccharomyces. So that's one reason. And then the second reason is that it's a Crabtree negative organism, so it doesn't ferment. Saccharomyces generates ethanol and kills itself at a certain point, right? And so with Pichia, we go to 400 grams of wet cell weight per liter here. So it's very high cell density. All right, thank you.
Thank you very much. We will have one last speaker in this session. I hope I can pronounce it correctly. Kivyon Cha, who is acting president of a company that he's going to explain. And
It's a public organization.
a public organization, okay. And the title is Coordinated Development for New Global Innovative Biotherapeutics in Korea. Please.
Okay, thank you. And then thank you for inviting me to this wonderful conference. Actually, I was the last person who enrolled this conference, and then I also thank you, Tom and Razinda, to squeeze in my session into this lovely conference. And also special thanks to two Gobins facing each other. Okay. Okay, to make the long story short, I brought very short video clips which I'm working on. My institute is a Korea government-funded public organization. So what we are doing is that you have done a lot of the mechanism-based research. So we call it mode of actions. What is going to be beyond that? So actually Nico and so many people has been introducing what's going to be the biotherapeutics after you are studying a lot of mechanistic work. So 20% of 30% when you go to the FDA to get the approval, 20% of and 30% is very important. Your mechanistic work is very important, okay? I cannot say it is necessary, but it is very crucial. Without it, probably 10 times harder time you will get it. But the leftover 70% to 80% is not something to do with the mechanistic. It is going to be safety issues, and then the glycosylation, and then there's processing issues. Okay, my institute is to solve that black box after your research, before the company is getting in. Okay? Oops, sorry. Okay. Can you make a voice? Okay.
Where does the strength that raises national competitiveness originate from? What do companies that dream of success need the most? Our inquiries meet the world like this. The place becomes the bridge to success for a medical industry and an economic growth in Korea. The new history begins in Osan Medical Innovation Foundation. The best way to predict the future is to invent it. Medical industry, the industry that the world pays attention as the next generation technology following automobile, IT, and shipbuilding. Noticing the value and infinite possibility of advanced medicine since the early 90s, Korea has started creating Korea's first medical complex, Osan Korea Medical Cluster, since 2009 as a part of National Project. We didn't pause at the first step. Settling the world's only R&D cluster where companies, universities, research institutes, and biohealth area, national institutions are concentrated. Osan Korea Medical Cluster has proved its potential. Osan Medical Innovation Foundation stands as the center of incalculable growth. Osan Korea Medical Cluster has experienced a rapid growth with an unstinting R&D support by Osan Medical Innovation Foundation. It is not a coincidence that global companies chose Osan Medical Innovation Foundation. Tomorrow belongs to those who have vision today. Osan Medical Innovation Foundation provides one-stop service for R&D. New Drug Development Center, which develops and optimizes new drug candidates to promote the development of new biomedical drugs. Medical Device Development Center, which supports all stages from the R&D of medical devices, production of test product, to trials in one stop. Laboratory Animal Center, which provides world-class system for preclinical evaluation of newly developed drugs and medical devices. And Clinical Drug Manufacturing Center, which manufactures biopharmaceutical products according to GMP. The 4 core research support facilities support the entire process from product development to commercialization. Also, through the Clinical Research Hospital that will enable clinical trial, it will become the world's only biohealth cluster that will support from the basic R&D support to commercialization in one stop. Located at the center of the nation, Osan Korea Medical Cluster will reduce your cost and time through outstanding transportation network based on its geographical conditions. Easily provide necessary information for R&D through national research support facilities. And 6 public health administrative organizations near the cluster offers more convenient licensing processes regarding products, including R&D and manpower training. Differentiated incentive reduced companies' financial burden and eliminated inconvenience in institutional support. The place that completes global-class infrastructure in medical industry from R&C support to licensing and commercialization. Osan Medical Innovation Foundation will complete Osan Korea Medical Cluster as the new role model of medical industrialization and lead companies' development.
오송 첨단의료산업진흥재단은 의료산업이 대한민국의 차세대 경제성장 동력이 될 수 있도록 글로벌 수준의 바이오 신약과 첨단 의료기기 연구로 개발을 지원하는 기관입니다. 저희 재단을 통해서 첨단 의료산업이 대한민국의 국부를 창출하고 대한민국의 국격을 드높일 수 있도록 하겠습니다.
I'd rather attempt to do something great and fail than to attempt to do nothing and succeed. And now we face the future again. We challenge towards the new tomorrow without rushing or giving up. At the place where state-of-the-art companies in Korea and worldwide chose and Korean government supports. As the global advanced base of medical industry that leads Korea's economic growth and job creation. To become a global biomedical hub that leads the world. Wait and see. The place where your success, Korea's growth, and global medical industry's success happen. This is Osan Medical Innovation Foundation. Osan Medical Innovation Foundation.
Knowledge city and then scientific city, and you are also planning for. And then 10 years ago, Korea has started to do this biocluster projects. Okay, $8 billion supposed to be invested to establish, and then right now the $1 billion has been already put it in. So not only all the building, now after groundbreaking, actually it's been 4 years to develop all the technologies to do it. So I'm not gonna overlap with the video clips. What I can do is something short talk without any overlaps. This is personal picture. This is a personal picture with the Govin and Esther over here. And yesterday I asked my wife. Okay, many people is showing their pictures. Let me please send me. Okay, some the picture with Govin, and then this is my wife's collection. It's not my scientific collection. So Govin and Esther is there, and then when I come to Boston, 1994, actually after 1 year working, we had my baby's first year birthday ceremony, and then I asked Govin and to invite, and then Govin and Esther was showing together. Then with my dad and the mom with my wife, okay, is there. So this is a lovely picture. So what also Govin is like a family father for me. Okay, this is again the this is my wife's collection. So the always with the kids. So actually, when my professor, I have two great scientists, and Govin and my previous supervisor, PhD supervisor, Professor Im. Actually, he visited with his daughter to the Boston, and then Govin invited us to the Rockport, and then had taken a picture. Actually, Esther has taken these pictures. So I also thanks to Esther as well as Govin. Okay, what we are doing is to support and promoting your mechanistic work, your basic science work, to go to the end of the clinical therapeutic development. Okay, this is all the overlaps. Korea has designated the Osan as bioclusters, like Chandigarh did for this knowledge city. So the FDA and then KCDC and the 6 government administration, the offices has been moved from the Seoul to Osan, and then they made a core infrastructure which I'm working on, K-Biohealth. The idea is that this is overlaps, new drug development center, medical device development center, and then laboratory animal center, and then the clinical drug manufacturing center, which has a biotherapeutics at the GMP level. Then we call it one-stop service. When professor coming or company people coming, no matter what the size of a company, if you have a targets, if you have a protein biotherapeutic, the hits, now we can optimize it before that can be injected into the human body. The Govin used to say, right, Kiwon, you have to make sure, you have to be very sure for your compounds, right? Same things goes on. We have to be very sure for biotherapeutics before we inject into the human body. Everything has to be done. Everything has to be resolved before we get the side effect from the patient. So that's why this one requires $1 billion per one product. 3 years ago, now they say $2.6 billion per project. That requires the biotherapeutic development. But once you are successful to do that, now you are gonna have 50 or the $70 billion can be obtained. Here is the problem. Okay, this is what you are doing in the academia and institute of R&D pipeline. You have your own ways, mechanistic ways, doses, and then this is what company is doing. Sometimes companies started from the R&D, and then this is for the commercialized drugs. There's huge gaps. What academia people is thinking, and the other company developing people is thinking. So Usually big pharma like Pfizer, Merck, and Sanofi or Novartis, they have their own specialists and their own apparatus and then technology to fill it up. No other company has it. And then unfortunately Korea, I don't think India, they don't have apparatus or technology to fill it in. So Korea government, actually 3 ministry comes together to solve this problem and then put some investment to solve it, to help the resources and then the professors and then company. This is the area what we are focusing on. Target validations and then mechanistic studies. Professor is doing so good job. I think all of you are doing a very nice job in your life. And then company is taking care of the last process of this drug development. There is a huge black box here. So only big pharma is doing globally, but Korea make a decision to. When you have targets, hit compound can be made by phage screening or protein engineering, okay? And then there is optimization process. No single product can be injected without optimization into human body. So very thorough process need to be done before that can be injected. That call it the lead compound identification and the optimization and candidate. Means, okay, I will do that. So this is something public area need to be done. The strategic plans and all the process and then technology can be explained whenever all of you guys can visit Korea or visit our institute. Many things. How the FC engineering, protein engineering for optimization. And then all the validation based efficacy evaluations. Okay. Mass spectrometry based evaluation, and then more or less a safety evaluation for the animals and cells, and then the immunogenicity, what we established for the glycosylation, those things, okay. And then process development to get the grams of protein out of liter. So with the Samsung manufacturing, they have 360,000 liter reactor with a productivity of more than gram per liter. Okay, so the other one I can explain whenever you have a time to visit us, Korea, and our institute. Okay, thank you. So everybody is hungry. Okay, thank you.
Thank you very much. I have one question.
Okay.
Who owns the final intellectual property?
Okay, very important question. So, right now we are giving the company. Basically, the basic idea is we are not making a problem before they have a final products, whatever intellectual properties or the other one. So far we are giving if they have their targets, we are giving the services, no intellectual property. But some small company like to share the profits, then we have reduced the prices to the
But
debt.
if you have your professor in this red image that you have there,
Okay,
and his or her graduate student,
I'm sorry?
his or her graduate student, can they publish their result?
Oh, yes, you can, but this special area, professors sometimes give up the publication. But there is some field we need to develop the new processes or the protein engineering field, then we can publish. But it's not like a mechanism based. So that's possible.
I'm really very impressed but also we have to develop new processes for intellectual property
Right,
publication.
right. Right.
I thank everybody who spoke this morning, and we are now going to having a picture taken, or where do we go?